This is Libby, and I'm going to be talking about testing for biological molecules today. This is the last topic within the enzymes section. Testing for carbohydrates, lipids, and proteins. Reagents can be used to test for the presence of various food substances. The first step is to grind up the food and add distilled water to dissolve some of the food. You can then test for the different food substances present. So proteins, to test for proteins, you add beerroot solution. It would turn mauve or purple if proteins are present. To test for lipids, you add Sudan-3, a fat-soluble dye. If lipids are present, a red-stained oil layer will float on the water surface. The presence of lipids can also be tested by adding ethanol. The solution is added to water and shaken. If the solution turns cloudy, lipids are present. Testing for sugar. So to test for sugar, you add Benedict's reagent and heat for about 2 minutes. It will turn any of green, yellow, or red if sugar is present. The color depends on the concentration of sugar. And finally, to test for starch, you add iodine solution. It will turn blue-black if starch is present. So, to recap what we've just learned, if we add protein to beerroot solution, it will turn, let's just think back on what we've learned, it's going to be turning mauve or purple. And we can use tests to identify the different types of biological molecule. And to test for starch, as we've just learned, iodine solution is added, and the starch will turn blue-black if it's present. So let's just recap on these food substances and what tests we do. We know for lipids, we add Sudan-3 or ethanol. For sugars, we add Benedict's reagent and heat. And then for proteins, we add beerroot solution, which turns mauve or purple. So that's everything for testing biological molecules. Thank you very much for listening.